Hello, everyone. Today we're starting Chapter 3, Cell Structure and Function. Everything we'll study about microbes, from how they cause disease to how antibiotics kill them, comes down to their structure and function. If you understand what each part of a bacterial cell does, you'll understand why some infections are so hard to treat and why some bacteria are more dangerous than others. These 4 words describe what all living things, microbes included, must be able to do. Growth, increase in size or number of cells. Reproduction, make more of themselves. Responsiveness, sense and react to the environment. For instance, bacteria may swim toward nutrients or away from harmful chemicals. Metabolism, perform all the chemical reactions that keep them alive, making energy and building cell parts. So when we talk about killing bacteria, antibiotics usually target one of these life processes, like stopping metabolism or reproduction. If a patient's wound culture shows bacterial growth, that's literally the first process here, growth and reproduction in action. This table summarizes which groups of microbes do which life processes. Even viruses are listed, but note they don't perform all 4. They can't grow or metabolize on their own. That's why viruses are not truly living. They hijack our cells to do their work. Bacteria and archaea, on the other hand, do all 4. They're independently alive. Mini check. Quick question, why don't antibiotics work on viruses? Because viruses don't have their own metabolism or cell walls to target. Here's a question for you to think about. The smallest free-living microbe, Mycoplasma, is non-motile. Why is it still alive? Pause the video now and see if you can answer it. Explanation. Movement isn't required for life. Mycoplasma can't swim, but it can grow, reproduce, respond, and metabolize, so it qualifies as alive. Clinically, Mycoplasma pneumonia causes walking pneumonia. Patients might not feel very sick, but it's stubborn because this microbe lacks a cell wall. That's why penicillin doesn't work against it. This figure shows different types of cells. The left one is Escherichia coli bacterial cells, and the one on the right is Paramecium, a single-celled eukaryote. Note the difference in magnification. Prokaryotes are bacteria and archaea. They lack a nucleus. Their DNA floats in an area called the nucleoid. They can read their DNA and make proteins at the same time. Super efficient. They don't have internal membranes. They are usually 1 micrometer or smaller, tiny compared to human cells. Clinical angle. Because they're so simple, bacteria can reproduce very fast, some double in 20 minutes. That's why infections can explode quickly. This image shows a typical prokaryotic cell. Prokaryote, simple, small, no nucleus. Pro means before, so prokaryote literally means before the nucleus. This is the classic diagram of a bacterial cell. Note, not all prokaryotic cells contain all these features. Key parts. Cell wall gives shape and protection. Cytoplasmic membrane controls what enters or leaves. Ribosomes make proteins. DNA nucleoid, genetic blueprint. Flagella or pili, movement or attachment. You don't have to memorize every label yet. Just start seeing how these parts work together like organs in a body. Analogy. Think of the cell wall as armor, the membrane as the security gate, and ribosomes as the hospital's pharmacy, where proteins, the medicines of the cell, are made. Eukaryotic cells, like those in humans, fungi, protozoa, and plants, have a nucleus and membrane-bound organelles. They're much larger, 10-100 micrometers, and more complex. Each organelle has a specialized job. The mitochondria make energy, the nucleus stores DNA, and so on. Clinical connection. Understanding eukaryotic structure helps us see why drugs can harm our cells too. For example, antifungal drugs can have side effects because fungal cells are eukaryotic, like ours. Here you can see the nucleus, our control center, and other organelles, mitochondria, Golgi apparatus, endoplasmic reticulum. All are surrounded by membranes, allowing specialized compartmentalized functions, something bacteria don't have. Question: which organelle makes ATP? Answer: mitochondria. This image helps you visualize scale. A chicken egg has about a 4.7 centimeter diameter. A red blood cell is approximately 7-8 micrometers in diameter. A parasitic protozoan such as Giardia is 14 centimeters in length. A bacterium such as Staphylococcus is approximately 1 micrometer in size. A virus such as orthopox virus has a 0.3 micrometer diameter. That's why we need microscopes to see bacteria and electron microscopes for viruses. Analogy: if a human cell were the size of a football field, a bacterium would be the size of a small car on that field, and a virus would be a marble. Another tell me why question to think about. Why did the scientists think Epulepicium fischelsoni was eukaryotic? What showed it's a giant bacterium? Pause the video now and see if you can answer it. Explanation: this microbe found in fish guts is so large you can see it without a microscope. The scientist assumed it must be a eukaryote because size usually means complexity. But under the microscope, it lacked a nucleus and organelles. Instead, it had DNA floating freely, just like bacteria. That's what confirmed it's actually a huge prokaryote. Clinical connection? Lesson here: size can fool you. When identifying microbes, what matters most is structure, not just appearance. Some bacteria secrete a sticky gelatinous coating called a glycocalyx. It's made of sugars and sometimes proteins. It's like a sticky coat that helps bacteria, first, stick to surfaces like catheters, teeth, or tissues, and second, avoid being eaten by immune cells. It's basically a slime coat that helps bacteria survive and cause disease. Analogy: if the bacterial cell is the criminal, the glycocalyx is the disguise, slippery and hard for police immune cells to grab. There are two main kinds of glycocalyces: capsule, thick, organized, firmly attached. Think of a bulletproof vest. Capsules protect bacteria from phagocytosis, being eaten by white blood cells. Slime layer, loose, water-soluble, sticky. Think of spilled syrup, helps bacteria attach to surfaces and form biofilms. Clinical example: the capsule of Streptococcus pneumonia is what makes it virulent. The capsule helps it hide from your immune system. Mini check: which one makes bacteria more slippery to immune cells, capsule or slime layer? Answer: capsule. This diagram shows both the capsule and the slime layer. Notice how the capsule looks neat and even, while the slime layer looks fuzzy and irregular. The picture also shows how the glycocalyx sits outside the cell wall. It's the outermost layer protecting the microbe. If you stain bacterial capsules in lab, they appear as clear halos around the darkly stained cells. That halo is the glycocalyx. Next, let's talk about flagella. First, we'll watch this animation on motility.
Motility is the ability of an organism to move by itself. The motility of a cell may enable it to move toward a desired environment or to flee from a harmful environment. Bacterial cells use structures called flagella, or in the case of spirochetes, flagella-like structures called axial filaments to enable this movement.
Flagella, the bacterial tail-like propellers. Not all bacteria have them, but those that do use them for motility, to move toward nutrients or away from harmful chemicals. Flagella are long whip-like structures that spin like tiny motors. Analogy: imagine a submarine propeller. That's exactly how flagella work for bacteria. Flagella have three main parts: filament, the long whip-like part that sticks out of the cell; hook, a curved connector that links the filament to the base; basal body, the motor. It anchors the flagellum into the cell wall and membrane and spins the filament like an engine. This structure allows the bacterium to drill through liquids. That's why motile bacteria spread so fast. Clinical note: motility helps bacteria like Helicobacter pylori move through stomach mucus and cause ulcers.
The long whip-like portion of the flagellum is called the filament. Filaments are composed of many identical globular protein molecules called flagellin. Molecules of flagellin form helical chains around a hollow core. Certain pathogenic bacteria can be identified and classified by differences in their flagellar proteins. In contrast with eukaryotic flagella, no membrane covers the filament of a prokaryotic flagellum. The whip-like filament inserts into a curved structure called a hook. The hook is composed of a different protein and connects the filament to the basal body. The basal body is composed of different proteins than the filament and hook. The basal body is composed of a rod and a series of either 2 or 4 rings, which anchor the flagellum to the cell wall and allow the flagellum to rotate 360 degrees. You're viewing a flagellum from a gram-negative cell. A gram-positive cell would only have 2 rings in its basal body, both of which are attached to the plasma membrane.
These slides show how flagella can be arranged differently. Monotrichous, one tail at one end. Lophotrichous, a tuft at one end. Amphitrichous, one or more at both ends. Peritrichous, all over the surface. These bacteria move like fuzzy ping-pong balls. Each arrangement suits a different lifestyle. E. coli is peritrichous, Vibrio cholerae is monotrichous. Mini-check. If you saw bacteria zooming quickly in all directions under the microscope, which type of flagella arrangement would you expect? Answer, peritrichous.
Cells with a single flagellum are monotrichous. When the flagellum rotates counterclockwise, the bacterium runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Amphitrichous bacteria have a flagellum at either end of the cell. Bacteria with a tuft of flagella at one end are called lophotrichous cells. When the flagella rotate counterclockwise, the flagella bunch up and the cell runs. When the flagella rotate clockwise, they separate and the cell tumbles. Peritrichous bacteria have flagella covering the surface of the cell. Runs occur when all of the flagella rotate counterclockwise and become bundled, while tumbles occur when the flagella rotate clockwise and separate.
Some bacteria, like Treponema pallidum, cephalus, don't have external flagella at all. Instead, they have axial filaments, flagella wrapped inside their cell wall between layers. When they spin, the whole cell corkscrews through tissue. This helps them burrow into places other bacteria can't reach, like between skin layers or blood vessels. Clinical connection? That corkscrew motion is what makes T. pallidum so invasive and hard to detect early.
Spirochetes are a group of bacteria that are spiral-shaped. They move in a corkscrew-like fashion, using flagella that do not protrude from the cell wall like typical flagella. Lyme disease is caused by a spirochete called Borrelia burgdorferi. Another spirochete, Treponema pallidum, causes syphilis. Spirochetes have an axial filament that lies between the outer membrane and the cell membrane. The axial filament winds around the outer edge of the cell. The axial filament is made up of a bundle of modified flagella called endoflagella. Rotation of the endoflagella causes the axial filament to rotate around the bacterium, which causes the spirochete to corkscrew through its medium. It is thought that this manner of movement allows the spirochetes to move more easily through viscous human tissues and fluids, such as mucus. Then they can easily attach to the cells below.
Bacteria use flagella for taxis, movement in response to stimuli. When flagella spin counterclockwise, the bacterium runs, moves straight. When they spin clockwise, it tumbles, reorients. This alternating pattern helps bacteria navigate their environment like a microscopic game of hot and cold. Analogy? Imagine you're blindfolded and smell cookies. You take a few steps, run, stop and sniff again, tumble, then adjust your direction toward the smell. That's bacterial chemotaxis.
Although flagella may appear to undulate back and forth like an eel swimming through water, they actually rotate 360 degrees, clockwise or counterclockwise. The basal body spins, much like the drive shaft of an electric motor, producing this motion. When the flagellum rotates counterclockwise, the bacteria runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Movement toward or away from a stimulus is called taxis. The stimulus can be light in the case of phototaxis or a chemical in the case of chemotaxis. In either case, the bacterium contains receptors which send signals to the flagella. In the case of positive taxis, or motion toward the stimulus, tumbles become less frequent when the cell moves toward the stimulus, and more frequent when the cell moves away from the stimulus.
Besides flagella, bacteria have smaller hair-like structures called fimbriae and pili. These aren't for movement, they're for attachment and interaction. Fimbriae let bacteria cling to each other or to surfaces, while pili, a special type of fimbriae, help transfer DNA from one bacterium to another. Analogy: if flagella are the legs, fimbriae are the hands, and pili are the USB cables for sharing information. This image shows fimbriae in action, short projections covering the surface. They help bacteria form biofilms, which are slimy layers of microbes living together on surfaces like catheters, teeth, and heart valves. This image shows biofilms, which, like bacterial cities, they share nutrients and protect each other from antibiotics. Clinical relevance: this is why hospital-acquired infections are so hard to treat. Biofilms make bacteria up to 1,000 times more resistant to antibiotics. Each bacterium can have hundreds of fimbria, like Velcro spikes, helping them cling to surfaces. Clinical example: Neisseria gonorrhoeae uses fimbriae to attach to reproductive tract cells. Without them, it couldn't cause infection. Mini check: what happens if bacteria lose their fimbriae? Answer: they can't attach, so they can't infect as easily. Pili, singular pilus, are longer than fimbriae but shorter than flagella. They're often called conjugation pili because they connect two bacterial cells and allow DNA transfer, a process called conjugation. That's how bacteria share traits like antibiotic resistance. Analogy: think of a pilus as a little tunnel. One bacterium sends genetic material through it to another bacterium. Clinical note: this is one reason antibiotic resistance spreads so fast in hospitals. Bacteria literally pass the resistance genes to their neighbors. This figure shows what pili look like under the microscope. They're single, long, thin extensions compared to the shorter fimbriae. Usually a bacterium has only one or two pili used like grappling hooks for conjugation. Mini check: how are fimbriae and pili similar? Both help in attachment. How are they different? Fimbriae for sticking, pili for DNA transfer. Here's a question for you to think about. Pause the video now and see if you can answer it. Fimbriae and pili are made of similar protein building blocks and share the same function, attachment. The pilus is just a specialized fimbria for gene exchange. Flagella, however, are completely different, built for motility, not attachment or DNA transfer. Quick recap question: if you were to stop bacterial conjugation, which structure would you target, fimbriae, pili, or flagella? Answer: pili. The bacterial cell wall is the strong, rigid layer that surrounds the cell membrane. It's like a pressure suit that keeps the cell from bursting when water moves in. Functions include giving bacteria their shape, rod, sphere, spiral, protecting from osmotic pressure, helping bacteria stick to other cells, being a major antibiotic target. Many antibiotics damage or block cell wall formation. Clinical example: penicillin works by preventing bacteria from building their cell walls, so the cell bursts and dies. This figure shows classic bacterial shapes: coccus, round; bacillus, rod; spirillum or spirochete, spiral; and arrangements like strepto, chains; staphylo, clusters. So streptococcus equals sign chains of spheres. This figure shows classic bacterial shapes: coccus, round; bacillus, rod; spirillum or spirochete, spiral; and arrangements like strepto, chains; staphylo, clusters. So streptococcus means chains of spheres. Staphylococcus means grape-like clusters. These names literally describe what they look like under the microscope. Tip: remember, staph infection looks like grape clusters, just like Staphylococcus aureus on a slide. The main ingredient of the bacterial cell wall is peptidoglycan, a mesh-like molecule made of sugars, NAM and NAG, and short peptide chains. Think of it like a chain link fence that gives structure but still allows nutrients to pass through. Two key types of bacterial walls differ by how much peptidoglycan they have. Gram-positive, thick peptidoglycan layer. Gram-negative, thin peptidoglycan plus an outer membrane. Clinical tie-in. Knowing the difference helps choose antibiotics. Gram-negative bacteria are often harder to kill because of that extra outer membrane. These figures zoom in on what peptidoglycan looks like chemically. The sugars NAG and NAM link together in chains. Short amino acid cross bridges connect the chains, making a strong, flexible network. If you break those peptide links, like penicillin does, the whole wall collapses and the bacterium dies. Analogy: imagine a brick wall held together by mortar. Peptidoglycan is the mortar. Antibiotics like penicillin dissolve it so the wall crumbles. Mini check. What are the two sugar components of peptidoglycan? Answer: NAM and NAG. Gram-positive bacteria have a thick peptidoglycan layer, which helps them keep their shape and retain the crystal violet stain during gram staining. That's why they look purple under the microscope. They also contain teichoic acids and lipoteichoic acids. Teichoic acids help stabilize the wall and anchor it to the membrane. Lipoteichoic acids help the bacteria stick to surfaces or host cells. In some gram-positive species, like Mycobacterium, the wall also contains mycolic acid, a waxy fat that prevents drying and protects against disinfectants. Clinical connection? That's why Mycobacterium tuberculosis can survive for months in dry conditions. Its mycolic acid makes it nearly waterproof. This figure shows the structure of gram-positive cell visually. The thick purple layer is peptidoglycan. Beneath it is the plasma membrane. There's no outer membrane. That's important because antibiotics like penicillin and vancomycin can easily reach and disrupt this wall. Mini check. What color do gram-positive bacteria appear after staining? Answer: purple. Gram-negative bacteria have a thin peptidoglycan layer but a second outer membrane outside it. That outer membrane contains phospholipids, proteins, and lipopolysaccharides, LPS. This extra layer acts like armor, protecting the bacteria from many antibiotics and disinfectants. The LPS part is especially important because the lipid A portion acts as an endotoxin. Clinical connection? When a patient with a gram-negative infection like E. coli or Pseudomonas is treated and the bacteria die, lipid A can trigger inflammation, fever, low blood pressure, and even septic shock. That's why these infections can make patients very sick very fast. This slide visualizes gram-negative cells. Gram-negative, thin peptidoglycan, two membranes, pink stain. The outer membrane of gram-negatives also has porin proteins, tiny channels that let nutrients and small molecules in but block large or harmful compounds. Analogy: think of gram-negative bacteria as a double-walled fortress, harder for antibiotics to breach. Some bacteria have no cell wall at all, like Mycoplasma species. Without a rigid wall, they can change shape, pleomorphic, and pass through filters that normally catch bacteria. Because they lack peptidoglycan, beta-lactam antibiotics like penicillin are useless against them. Clinical connection? This explains why Mycoplasma pneumonia, which causes walking pneumonia, must be treated with drugs that target ribosomes instead, like macrolides or tetracyclines. Analogy: if most bacteria wear armor, a cell wall, Mycoplasma goes into battle wearing a soft hoodie, flexible but resistant to certain weapons. Here's a question for you to think about. Pause the video now and see if you can answer it. Back to the earlier diagram of a bacterial cell. It showed a thick wall and no outer membrane, so it's likely gram-positive. If it had an extra outer layer, it would be gram-negative. Mini check. When you think of gram-positive bacteria, what color do you imagine under the microscope? Answer: purple. Right under the cell wall is the cytoplasmic plasma membrane. It's made of a phospholipid bilayer, two layers of fatty molecules with proteins floating in it. We call it the fluid mosaic model. Fluid because the parts move around, mosaic because it's made of many different molecules. Analogy: Think of it like a flexible skin with floating doors. The proteins act as gates or pumps to let certain things in or out. Clinical note: this membrane is essential for maintaining balance inside the cell, so if a drug or disinfectant damages it, the cell leaks and dies. This figure labels the bilayer, showing the phosphate heads, water-loving, on the outside and fatty acid tails, water-fearing, inside. You can also see the integral proteins that span the whole layer and the peripheral proteins that sit on the surface. These proteins transport nutrients, send signals, and maintain energy gradients. Clinical connection: in nursing, this concept connects to how disinfectants like alcohols and detergents kill bacteria. They dissolve these membranes.
The cytoplasmic membrane of a prokaryotic cell is the main gateway for movement of molecules in and out of the cell. This membrane may also be called the cell membrane or plasma membrane. Cytoplasmic membranes in bacteria are comprised of a phospholipid bilayer. Each phospholipid contains a phosphate head located on the outer part of the bilayer, attached to a glycerol and 2-fatty acid, which face the core of the membrane. The phosphate is negatively charged and hydrophilic, while the tail portion of the phospholipids is hydrophobic. Thus, cytoplasmic membranes are very effective at blocking passage of many molecules, while other molecules are allowed to pass. Cytoplasmic membranes also contain two types of proteins. Integral proteins often span the entire bilayer and act as channels for transport of molecules that normally wouldn't be able to penetrate the bilayer. Peripheral proteins are loosely attached to the membrane on one side or the other. Some of these may be glycoproteins, which are proteins bound to a polysaccharide. These proteins may function as recognition sites, enzymes, or receptors.
The membrane's job is to control traffic in and out of the cell. It's selectively permeable, meaning only certain molecules can cross. It also stores energy, like a battery, helps with ATP synthesis, maintains a difference in charge across the membrane, called the electrical gradient, that drives movement of ions and nutrients. Analogy: it's like hospital security, controlling who enters, who leaves, and keeping the electrical systems running smoothly. Mini-check: what term describes the idea that the membrane allows only certain substances to pass? Answer: selectively permeable.
With their hydrophobic cores and charged surfaces, cell membranes are very efficient at blocking passage of many molecules. Some molecules, such as water, dissolved oxygen, and simple alcohols, are able to pass through cell membranes freely by passive diffusion. However, charged molecules and large molecules cannot pass directly through cell membranes, and hydrophobic molecules tend to become stuck inside the hydrophobic interior of cell membranes. In order to allow these complex molecules to enter and exit the cell, organisms rely on two forms of membrane transport: passive transport and active transport. Passive transport occurs along an electrochemical gradient. It does not require the expenditure of energy in the form of ATP for transport. Active transport requires the expenditure of ATP in order to transport molecules across the membrane. Active transport and some forms of passive transport require the use of transport proteins, such as the integral protein seen here.
This slide shows how the inside of a bacterial cell usually has a negative electrical charge compared to the outside. This happens because of the uneven distribution of ions like sodium Na+ and potassium K+. That electrical gradient helps drive transport, kind of like water behind a dam. The cell can use that stored charge energy to move molecules and make ATP. Mini-check: what's the term for the stored energy created by the charge difference across the membrane? Answer: electrical potential or membrane potential.
Passive transport includes diffusion, nonspecific and specific facilitated diffusion, and osmosis. All of these processes occur along electrochemical gradients and do not require the expenditure of energy in the form of ATP. Diffusion is the net movement of a chemical down its concentration gradient, that is, from an area of high concentration to an area of lower concentration. This diffusion requires no energy expenditure by the cell, as the concentration gradient drives the diffusion. Small uncharged molecules, such as dissolved oxygen, dissolved carbon dioxide, small alcohols, and small fatty acids, can freely diffuse through the cytoplasmic membrane. Once equilibrium is reached, molecules will still move across the membrane, but there is no net movement in a particular direction.
Now let's talk about how materials cross the membrane. The simplest way is diffusion, when molecules move from where there's more to where there's less. It doesn't require energy, it's a natural process. Examples: oxygen entering a cell, carbon dioxide leaving a cell. Molecules just drift across the membrane until they reach balance. Analogy: think of perfume spreading in a room. You spray one corner and soon you smell it everywhere. That's diffusion. Second, facilitated diffusion. Larger molecules need a doorway, a protein channel or carrier, to help them cross. Third, osmosis, the diffusion of water through a membrane. Water moves toward the side with more solute stuff dissolved in it. Clinical link. In IV therapy, understanding osmosis is key. Give too strong or too weak a saline solution and cells will either shrink or burst. This figure shows the three types visually. Molecules passing directly, simple diffusion. Molecules passing through a helper protein, facilitated diffusion. Water crossing through special channels called aquaporins, osmosis. Notice that all arrows go from high concentration to low. That's the key idea. Mini check. Does diffusion require ATP? Answer: no. Osmosis specifically refers to water movement. Water always moves from where there's more water, low solute, to where there's less water, high solute, trying to equalize both sides. So if the outside of a bacterial cell has lots of salt, water will leave the cell and the cell shrinks. Analogy: imagine a raisin soaking up water and swelling, or in reverse, drying out and shrinking. That's osmosis. This is one of the most important clinical slides. It shows what happens to cells in different environments. Isotonic, equal concentration in and out, cell stays the same. Hypotonic, more water outside, water moves in, cell swells and might burst if no wall. Hypertonic, more solute outside, water moves out, cell shrinks, plasmolysis. Bacterial cell walls help prevent bursting, but human cells can't handle this as well. Clinical connection. This is why IV fluids are isotonic, like 0.9% sodium chloride. Too much or too little salt would damage red blood cells.
Simple diffusion does not require special membrane proteins for movement of molecules. However, other types of diffusion do require such proteins for the transport of molecules. Nonspecific facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of nonspecific facilitated diffusion, these permeases allow a wide range of molecules that meet charge or size criteria to pass through their channel. Some facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of a specific facilitated diffusion, these permeases only allow specific types of molecules to pass through their channel. The channel contains a recognition element. If the molecule is not recognized, the channel will not open to allow the molecule to pass into the cell. Osmosis is a special kind of passive diffusion that involves the movement of water molecules across a semi-permeable membrane. Water passes through the phospholipid bilayer with some difficulty, but transmembrane channels can ease the passage of water through the membrane in a process called facilitated diffusion. Water plays a critical role in maintaining cell integrity. Water tends to move across the membrane from areas of high concentration to areas of low concentration, just like solutes. The difference is that water makes up most of the liquid, so a change in the total concentration of solutes is really the force that causes water to move. If the concentration of solutes outside the cell is high compared to the concentration inside the cell, water may leave the cell, causing the cell to shrivel up. Similarly, if the concentration of solutes inside the cell is high relative to the outside, water may move into the cell and can cause it to swell up and possibly burst.
Now let's switch gears. Active transport requires energy, ATP, because the cell moves materials against their concentration gradient from low to high. It's like swimming upstream. Active transport allows bacteria to take in rare nutrients even when they're scarce outside. Analogy? Think of it as using a pump to move water uphill. It takes energy, but it lets you store what you need.
Active transport requires the cell to expend energy, usually in the form of ATP, to transport materials across the cytoplasmic membrane. This transport occurs through transport proteins and is usually against the electrochemical gradient, like carrying a load uphill. There are three types of transport protein. Uniport proteins transport only one type of molecule. Antiport proteins transport two types of molecules but in opposite directions. Symport proteins transport two different molecules at the same time but is coupled with a uniport channel.
This figure shows the main types of active transporters. Uniport, one substance, one direction. Symport, two substances move together in the same direction. Antiport, two substances move in opposite directions. All require energy to push molecules where they wouldn't normally go. Clinical relevance. Some antibiotics and antiseptics work by blocking these pumps, starving bacteria of essential nutrients or ions. Mini-check. Which type of transport can move molecules from low to high concentration? Answer: active transport.
A uniport is an active transport protein that transports a single molecule or a group of the same molecules in one direction against the concentration gradient. These proteins are also ATPases, using ATP to drive the transport. An antiport is an active transport protein that transports two molecules in opposite directions against their concentration gradients. These proteins are also ATPases, using ATP to drive the transport. An example of an antiport protein channel is a sodium-potassium gated channel that maintains the electrochemical gradient of cells, which is responsible for maintaining a voltage difference between the outside and the inside of the cell. A symport is an active transport protein that transports two different molecules across the cell membrane at the same time. In this case, it's coupled with a uniport channel that uses ATP to actively transport a molecule against its concentration gradient. When those same molecules are in high concentration outside the cell, their diffusion back into the cell provides energy to transport a different molecule against its concentration gradient through the symport.
There's one special type of active transport that only prokaryotes bacteria use. It's called group translocation. Here's how it works. When a molecule like glucose enters the bacterial cell, it's chemically modified, often by adding a phosphate group. That modification traps it inside the cell because it's now a different molecule. Analogy? Imagine walking through an airport gate that stamps your passport. Once you're inside, you can't use the same passport to get back out. You're now changed in the system. Clinical note: this helps bacteria hoard nutrients. It's also why some antibiotics target bacterial enzymes that assist in these energy-based transport systems. This diagram shows the process step by step. The molecule, like glucose, approaches the membrane. Transport proteins use energy to pull it in. Enzymes modify it as it enters, such as by phosphorylation. The result: the bacterium gains food and keeps it locked inside. Mini-check. What makes group translocation different from other active transport? Answer: the substance changes chemically during transport. This table summarizes everything we've discussed about transport: passive diffusion, facilitated diffusion, osmosis; active, uniport, symport, antiport, and group translocation. You can think of them as a spectrum. Passive means no energy, moves downhill. Active means uses energy, moves uphill. Tip: for nursing students, the same logic applies to human physiology, like how our kidney cells use active transport to retain sodium in water.